Hi everyone, welcome back to Biology 101. This is Amrita and today we have another topic, that is codominance. So till now we were discussing crosses where the F1 progenies resembled either one of the parents, that is dominance, or the progenies were in between the characters of the parents, that is incomplete dominance. Example of incomplete dominance we saw was the snapdragon, pink color snapdragon flower, right? But now in this case of codominance we have, that is first generation will resemble both the parents. A blood group of the human body, that is ABO blood grouping, is a very common example of codominance. Now this ABO blood groups are controlled by I gene, right? I gene, the plasma membrane of the red blood cell. There is a red blood cell. Now this is the plasma membrane. Now the plasma membrane of the red blood cell has sugar polymers. Let's say this is a sugar polymer. Sugar polymers that protrude from the surface, and the kind of sugar is controlled by the gene. Now let's say this I gene has three alleles, right? IA, IB and small I. These three are the alleles are what? Basically different forms of a gene. So I gene means IA, IB, small I. These three are the alleles of the gene I. Now the alleles IA and IB, they produce some type of sugar, but this that is small I, does not produce sugar. Okay, does not produce sugar. Now because humans are diploid organism, each person possess any two of the three alleles, that is it can be IA, small I, IB, small I, anything can be. Now we will see all that. Okay, so now IA and IB, okay, the IA and IB are completely dominant. These both are completely dominant, and this is dominant. Okay, so this small I is recessive, right? That is dominant and this is recessive. Now when IA, for example, I said, right, if IA and small I is there, then the blood group will be A, that is IA only expresses itself because that is dominant. If B and small I is there, B, because IB is dominant, so it expresses itself. Small I cannot express because that is recessive. Now red blood cells have, if it is IA and IB, that is IA, IB together means it will produce AB blood group, and it will have both the type of sugars, that is A sugar also, B sugar also. Since there are three different alleles, and so we can have 6 different combinations of these alleles. So let's write what are the alleles. Now this is from parent 1 we have, and from parent 2 we have the alleles. We will write here the genotype, phenotype. Okay, so here from parent 1 we have IA, let's say. From parent 2 IA, I am writing all the combinations possible. IA, IB, IA, small I, okay, small I, then IB, IA, IB, 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 small I, the final one small I, small I. So first one we know IA means the phenotype will be A because both are dominant. IA, IB, again both dominant, so AB. IA and small I, we have A because A is dominant. IA is IB, IA, or IA, IB is same, so we have AB. Now IB is B because B is dominant. IB and small I and B again. Now II we have O. So from here 1, 1, 2, 2, 3, 4. So we have 4 phenotypes and 6 genotypes. IA and IB and IA, IB and IB, IA are same. So we have 6 genotypes and 4 phenotypes. Okay, so this is basically codominance. Now let's recap. See, basically codominance is a deviation from the normal dominance, that is it can possess both the characters of the parents, that is basically codominance, and we have a very good example, ABO blood grouping in the human bodies. from that blood grouping we are just putting out examples, like if IA and IB come together, we'll have AB. If otherwise, it's normal dominance. This IA IB, this part IB small I, IA small I, we have A here and B here, right? This is normal dominance. But this IA IB and IB IA, these both are examples of codominance. So we are just differentiating between the dominant one and the codominance one. So thank you. We'll see next multiple allelism in the next video. Thank you for now.